We start our lecture, the concept of transcription factors and signaling pathway. Lecture number 3. And first, I need to say about every lecture or every cells have communication between each other, between tissue, between different cells, and it will paracrine, endocrine, and autocrine way. All this way work through signal mechanisms, and this signal mechanism has a structure which present on this slide. It is first receptor on membrane. A receptor need to connection with ligand or a primary messenger. Then will signal start come inside of cell and start to work signal transduction via second messengers. And this signal can stay and cytoplasm and give cellular responses, or come to nucleus and activated transcription factors. These transcription factors will work with DNA and change gene expression, and it will make protein production after translation and also give cellular responses. These rules work for all cells, and in our cells present many signal ways which can make cross between different ways or work separately. And what we need to know about type of receptors. Receptors may be very different, and these differences may be from localization. It will be membrane or cytoplasmic. If membrane, so ligands need to work outside of cells. If cytoplasmic, so ligand or primary messenger will come to cells and will work inside of cells. Next type of receptors will be different by function. It may be tyrosine and serine threonine. It is enzyme which make activity. More often it is phosphorylation or dephosphorylation, and make activity or inactivity second messenger, and signal will go to next step. What we need to know about biological function. The first, receptors that induce signal for cell survival and proliferation. The second, the receptors that induce cell differentiation. Today we know or have information about more than 200 types of receptors, activate about 30 intracellular signaling pathway, and several hundred transcription factors that regulate about 20,000 target genes. It is very important information because in every of these receptors or protein which includes in signal pathway or transcription factors may be mutation, and this mutation will change all information about transcription of genes. What you need to know about ligands. This is a biologically activity substance. It may be drugs, it may be hormones, neurotransmitters, mediators, growth factors, and it is all that give information for receptor. They work, come to receptor connection. It is on our slide, step number 1. Receptor change structure and can activate, and result of this is activate will example ion transport or enzyme activation or protein synthesis. And then step number 2, ligand need go away, and receptor will return to his first conformation or shape, and again ready to connection with next dose of ligand. So if regulation system work in high activity and need to make a cell response very high or very fast, ligand will produce many, and they will fast work with receptors and repeats, and will activity and response of cells for this is ligand. What means signaling pathway? The main type of modification signal transduction proteins in their phosphorylation and dephosphorylation. I said about previously. Depending on the site of the protein undergoing phosphorylation, this reaction can cause opposite effect, activation of the protein or their inhibition. In this slide, you can see most famous signaling pathway. Now, here presented only 5 pathway, but in real it is more. I said previously more than 100, and they can work crossed. Example, one signaling pathway can give information for another, and they can activate it, regulate it, and inhibition another pathway. It make very strong and very difficult cell-cell interaction and signaling pathway and cell response. In this slide, you also can see special position which we discussed previously. It is ligand, receptor, effector or proteins which includes in signaling pathway, and target. This is target may be transcription factors which will sit on DNA, and activation or inhibition gene transcription. So some of gene we will call or label as target genes. And when we will speak target genes, you will understand that transcription factor can activate it or inactivate it, special genes which promoter space this is transcription factor can to know and sit and activate it. And next part of lecture, we will speak about some of signaling pathway and to understand how it works. And first, it will notch signaling pathway. It is very often and very famous and very important to know in leukemia, disease when blood cells will tumor cells, and notch signaling pathway will work high, and it give many new tumor cells which will foundation from blood cells, B lymphocytes or T lymphocytes. And here you can to look ligand and receptors, and when ligand and receptor meet, they make new conformation or shape, and signaling will come to cells. Result of this is activation signaling will activate transcription factors, which will sit on promoter of gene E and make transcription position on or start of transcription genes. And second situation or position off when ligand not come to receptor and signaling way not work. So transcription factor not have co-receptor or activator of transcription factor come blocked factor, and transcription of gene will in position off, transcription factor will degradation, and target genes not will transcription. On this slide you can to look some photo of some patients which has mutation in this system, and Notch1 receptor with mutation not work as need, not work normal, and this is mutation we can to meet in more than 50% of cancers. And more often it will T lymphoblastic leukemia, chronic lymphocytic leukemia, diffuse large B-cell lymphoma, mantle cell lymphoma, breast cancer, and non-small cell cancer. It means some of signal pathway maybe has special tissue effect. Example here, many disease which character for blood cells and leukocyte cells. But also it may be as a contrast, notch signaling will inactivate it. Also maybe mutation which will not active, which will blocked. Example in carcinoma happens in skin squamous cell, head and neck squamous cell, esophageal squamous cell carcinoma. Again, we speak about mutation, make or activation or inactivation, that it will happen in special cells and give special disease. Next, transcription factors, TGF-beta or tumor growth factor beta. And also of this is slide you can see scheme, traditional scheme for signaling ways. It is ligand which will connection with receptor. It is some of proteins which will work after connection with receptor and ligands. some proteins which will phosphorylation or dephosphorylation and action as second messenger and give information for nucleus, for transcription factors, which will take place in DNA promoter zone and in result have activation of transcription target genes. And here you can to see that in cells present inhibition factors Smad7 which can make block and signaling node will come to nucleus, so node will affect. Understanding about activation factors and inhibitor factors very important because we can use this is biological molecule for building drug construction. We can make same drug which will inhibition or which will activation, and we can from outside input inside of cells any molecule, any drug to regulation effect which need for treatment of disease. And also you need to understand that mutation in receptor zone, mutation in every of this is protein, mutation in inhibitor, or a mutation in enzyme which will make phosphorylation, dephosphorylation, will disturb normal work of signaling way, transcription factors, and disturb gene expression. And next slide, you can to see different polymorphic of proteins, polymorphic of receptor, TGF-beta. It is one polymorphic TGF-B receptor 2 second type and TGF-B or receptor first type. They have different effect. They can make different biological function. Example, receptor type second type can make block development, can make block proliferation. So if we will activation this is receptor, we will not have development or proliferation. And receptor number first or first type can induce the differentiation and maybe here it is scientific research will show in future, maybe can regulation proliferation. So if this is receptor will work, will this effect, if not will work, so this is effect not will work also. And in right side you can to look Smad7, Smad4, Smad2, Smad3. It is proteins which can regulate it effect of receptor and ligand TGF beta 1 and TGF-B receptor first and second type, and can connection with cells and make activation development, activation proliferation, activation apoptosis, activation dedifferentiation or any function or block. And also I need to repeat information. If we will understand this is scheme, we can make LEDs. or regulated of tumor cells and make what we need. Example, in last lecture I told you about tumor biology of cells, and I mainly speak about apoptosis, which will down regulation in cells, and many cells will with mutation not die and give many tumor clone, but if we will activation apoptosis, this cell will die and tumor not will development. No, also slide which can make more can show for you more difficult how it real work, how many proteins work, how they work. They always make homodimer, dimer, maybe connection, disconnection, and from this combination will make action or not action. And also you can to look here ligands which outside of cells come to receptor. Receptor, it is a very difficult structure which have outside part, intramembrane part and inside part. And all this is proteins has many function, and this is function work in any case and give different effect. Example, one effect with phosphorylation and connection with Smad2 will give protein expression, because this is shape can to come in nucleus and make sit on promoter and can make target genes expression, transcription. But if this is proteins will work with SMAD3, it will block of effect and not come to nucleus. So genes not will transcription. And also we need to understand that receptor and ligand also can regulation. If we will use ligand which have shape same ligand but not make biological function, so it not will work in cells. Receptor will busy with this new ligand, but function not will come to cells. It is also can use for regulated all effect of cells. And if I said in last lecture that tumor cells has many blood for catch many growth factors, many hormones. So here, if we will use fake ligand which not give biological effect, it means we will block for tumor cells any biological molecules which these tumor cells use for growth. So growth will inhibition and tumor will go away. No, this is new pathway, signaling pathway. It is VNT beta-catenin of pathway. Some of you in course of big data in biology research beta-catenin protein, and you know about different polymorphic proteins, about polymorphic genes which can happens as result as mutation, and how it can detection. And now you can to see what it need to know about beta-catenin. And look of this slide, you can to see also VNT beta-catenin pathway has special structure. It is receptor which has outside ligand or outside part, intramembrane part, and inside part. It is many proteins which includes in this way APC, also was protein. In big data we speak about polymorphic protein and polymorphic of genes of this protein, and means this is protein which includes in this signaling way can have many mutation and can not work very excellent. This way has ligand VNT, which connection with his receptor make this is signaling pathway on state. What will in result? In result, after ligand and receptor connection, beta-catenin will come to cells and will separate. Then this is protein will come in nucleus. Now this is beta-catenin will plays role as second messenger and transcription factors. He need to no he not special transcription factors. He helper of transcription factor. You see green on slide, you see green part which on DNA they connection with beta-catenin. Catenin help to activation, this is transcription factor, and will transcription VNT triggered genes or target genes and will cell proliferation. So if we have many VNT, we will have as a result cell proliferation. If we have any mutation in APC, exin, or beta-catenin, and they will work without VNT but will give effect of transcription factor activation, we will have many cell proliferation. And also if we will make block every step or one of step, we can regulation cell proliferation. Next slide can to show to us VNT off state. It means when signaling way not work and target genes of this is VNT pathway not expression and cell proliferation not happens. And here you can to see VNT or ligand not come, not connection, primary messenger, not connection with receptor. So beta-catenin in phosphorylation position and cannot separate from this complex. So they will proteasomal degradation not come to nucleus, not helps to transcription factors, and not will transcription. So it means we can block VNT, which previously give gene expression, or we can to use any drug which will block this receptor and not give dephosphorylation activity for any proteins, or we can use any other blocked for this position for catenin not will separate. This is need to remember, keep in memory, because we will use this in future when we will speak how we can regulation every of this pathway. No, and here a slide can to show to us how it may be used for real therapeutic pathway. and many researchers work and try to find any sites for make drug and for regulation. This is transcription way. Example A, VNT soluble receptor OMP 54F28. It is make block of VNT and not happens anything. B, same effect. It will Friesland antibodies, which will as a fake ligand work with receptor and not give anything. C, I find C, COX inhibition, and our beta-catenin was dephosphorylation and make separative shape and can come to nucleus, but this is type of drug, COX inhibitors, will not give to come for catenin inside of nucleus. Example D, it is TAN-kinase inhibitors. For make activation phosphorylation enzyme for separate beta-catenin, need that TAN-kinase enzyme which activate next enzyme will again in active shape. But if we will use any molecule or any drugs which will inhibitors, this is enzyme, so not will activation, not will activation, not will dephosphorylation, not will separative beta-catenin shape. Next, F. Also, here present way when VNT come inside of cells and will regulate it, and after his effect go away from cells to make new effect with receptor. But if make block this is effect, it will not give any results, and VNT will in cells, not will go outside, so not will work with new cells. And G, also it is inhibitors which need to come in nucleus, and also will regulate alternative splicing of transcription factors which need to connection with beta-catenin, with helpers. So if will another shape polymorphic of proteins, so it not will connection with beta-catenin, and effect of cell proliferation again not will happens. Result of this is 3 slides, from A to G. You need to understand every step can be regulation, every step can or activation or block, and result of this is regulation activity will result of effect of cell responsible. And you as a doctor or you as a researcher man can use this knowledge or use this knowledge for treatment or use this knowledge for make new drug and make experiment and make new effect of cells. And as example, you need to know that more than 90% of colon cancers are caused by mutation in the APC protein, which under normal circumstances target the degradation of beta-catenin. Hyperactivation of the VNT beta-catenin cascade in hepatocellular carcinoma is associated with protein gene mutation. 37% of cases it will with beta-catenin, 10% cases it will in exin protein, and less than 2% will in APC adenoma polypose colon genes proteins. It will happens with this is protein. And also you can to look that this signaling way can be used in different organs, in different tissues, and more special information for you. This is disturb or damage this is signaling pathway not will only with tumor. It may be different disease. About tumor I speak because it is only one model, but in model for explanation. But in real, same activity or inhibitor of activity can work in different disease. It is maybe COPT, lung inflammation disease. It may be bone inflammation disease. It may be skin and chronic wound which not have reparation and always present wound. It may be skin vitiligo. It is a spot, different color on hand, on face. It is maybe brain and disease which connection with blood good circumstance or not good. Ischemia, cerebral ischemia. And again, I need to show you transcription factor for memory. And again, I need to say that all signal ways have special but same structure. Ligand, receptor, effector, target. And next effector, we will more to speak about transcription factors. What is it and how it work. Transcription factors, it is proteins that bind to DNA regulatory sequence, enhancer or silencers. So it is regulatory activity which can make high gene expression or down gene expression, usually located in the fifth and upstream region of target of genes, and modulate the rate of gene transcription. I said about this previously. This may result in increase or decrease of genes. Transcription, protein synthesis as a result of gene transcription, and subsequent alterated cellular function. It is also, we was speak previously, it is a result of cell effect of hormone, drug, and any factors which can use cell as responsible target. Several families of transcription factors exist, and members of each family may share structural characteristics. These families include helix-torn helix, helix-loop helix, gene finger, example glucocorticoid receptor, GATA proteins. All this we was listened previously. It maybe was used in your previously subject. Next, basis protein leucine Z pair and B sheet motifs. Or example, nucleus factors kappa B also was used in our previously subject. Next, transcription factors may be two types. It is basal and regulatory. What means basal transcription factors? It is specialized proteins or proteins complexes whose function is to recognize gene promoters and form the pre-initiation complex polymerase second type. It will transcription factor second A, transcription factor second B, transcription factor second D, transcription factor second E, transcription factors second F, and transcription factor second H. Another type, it is regulatory transcription factors. It is recognized as a specific sequence of DNA sites bind in the regulatory region of genes. And this means that this is regulatory transcription factors need not work always in cells. They will work in any cases when need to cells make any activity or get any response. How it will looks. Here basal transcription factors, and you can to look of this slide TF, and they have enhancer and they have cofactors which connection with transcription factor TF and make more excellent activity of polymerase second. Polymerase second will make transcription and will give to new information how need and when need make gene transcription. All this activity we will to know as a transcription cycle, and here we'll has 4 position. The position number first, formation of pre-initiation complex on gene promoter and initiation of transcription. It is position number here, polymerase, transcription factors, and begin initiation. And this is complex will pre-initiation. Then will early elongation. This is complex make more activity polymerase second type, and they start to work and will early elongation. This is type. Then step number 3, productive elongation. Polymerase can also have helper, more same than was initiation factors, and this is processing factors will make more good and more long work of polymerase second. And next step, transcription termination. Polymerase will connection with 3 and factors which will make stop of polymerase second activity, and transcription will end, will finish. Transcription factors function in the nucleus where genes are found. It is normal. And nuclear transport, import or export of transcription factors can influence their activity. Another important general mechanism controlling the activity of transcription factors is post-translation modification, such as phosphorylation. Finally, in the addition to controlling the genes and transcription of the transcription factors, this protein complex can also control the genes responsible for their own transcription, leading to complex feedback control mechanism. In this slide, you can to look transcription factors families involved in the pathogenesis of asthma or another inflammation disease of lungs and esophageal structure. You remember I said previously that signaling weight and transcription, it is not only about tumor cells. It is also about many disease, chronic inflammation disease, and degeneration disease, metabolic disease, and so we use tumor only as example. In this slide, you can look glucocorticoids receptor. He can work as transcription factors. Nuclear factor kappa B, activator protein 1, cyclic AMF responsible element binding protein, signal transducer and activators of transcription, nuclear factors of activated T cells, gate binding proteins, hypoxia inducible factors number 1, CAAT enhancer binding protein, and specificity protein number 1. And more of them will so to know, special to know from big data when we speak about polymorphic of proteins, polymorphic of genes, and how it can detection. What next? Next, many transcription factors are common to several cell types, ubiquitous, such as AP1 and NF-kappa B, and may play a general role in the regulation of inflammatory genes. Whereas other are cell-specific and may determine the phenotypic characteristic of a cells. And next, you can to see NF kappa B signaling in inflammation. And here you also can to see that receptor which has outside part, intramembrane part, inside part. And you also can to look ligands which will connection with this is receptor. Then you can to look signaling weight from many proteins which will work as second messenger and give information for next proteins and activation inhibitor of NF-kappa B, and this is NF-kappa beta, kappa B will activate it, go to this is will go away, inhibitor will go away, and free of NF-kappa B will come to nucleus and will work as transcription factors and give gene transcription of special dependent genes. And as proteins which target of this is transcription factor will be non-activity shape of interleukin first 18 which will activate it and go away from cells and make any regulatory effect for inflammation in outside and give information for new cells. It will intermediation between different cells. And also you can to look that if many proteins includes in this way, so first maybe many mutation which can make another effect, and also it may be many regulation and can use many drugs which will make block or make activation. Example, this is need for make free NF-kappa B, need activated this is shape which will degeneration this is molecular. We can block here, block here, block here, block here. So many target for therapy, many target for regulation total effect of cells. In result, This is molecular, which will give cell responsible, which go away in fast and many, or no will come from cells and not will give any effect. About cross-talk. One of the most important concept I spoke about this previously, but again need to repeat. To have emergence is the demonstration that transcription factors may physically interact with each other to form homodimers and heterodimers, resulting in inhibition or enhancement of transcriptional activity at a site distinct from the consensus target for a particular transcription factor. This then allows crosstalk between different signal transduction pathways. At the level of gene expression, generally it is necessary to have coincident activation of several transcription factors in order to have maximal gene expression. May explain how transcription factors that are ubiquitous may regulate particular genes in certain types of cells. And here you can look again for different transcription factors which have any effect and can come cross-talk. And some more examples of disease which connection with signaling pathway dysregulation and transcription factor damage of work. And example first, it is 14-year-old male with PROS. And photo number A, it is frontal whole body view shows right facial, leg and bilateral feet hemihyperplasia, right more than left, after toe and metatarsal amputations and left abdominal fibroadipose hyperplasia. B. Facial view shows right facial hyperplasia and epidermal nevus involving his right cheek and neck. C. Lymphovascular lesion involving his right arm and trunk. Next example, it is about 30 years old male with Proteus syndrome. And photo number A, it is cranial hyperostosis involving his right frontoparietal bones. B, bone overgrowth with ulnar deviation of his bilateral second fingers. C, lateral view of his right leg shows bone hyperplasia and vascular malformation venous varicosities. D. Frontal view of his legs shows venous varicosities, bone overgrowth of his right leg. E. Dorsolateral view of left foot. F. Plantar view of left foot with cerebriform connective tissue nevus. Next example, 9 months old male, infant with a mosaic shape single nucleotide polymorphism in position 5390 when cytosine will change for thymine. You can to look how it label in real. Next, it is about position of amino acids. This is type of mutation in mTOR signaling way will associated with megalencephalia, development delay, dysmorphic features, mosaic pigmentary skin changes, and polymicrogyria. And also example about 20 years old male with PTEN Hamartoma Tumor Syndrome. He has a heterozygous when proline will change for histidine in position 38 variant in and associated with extreme macrocephaly, dysmorphic features, pigmentary skin, macula, development delay, and intellectual disability. And last example, a female with TSC, diagnosed based on renal angiomyolipomas, angiofibromas, all this is destruction with vessels, and hypomelanotic maculas, has congenital overgrowth of the right thumb and forearm that may represent type 2 segmental mosaicism. Now my lecture ends. Thank you for attention and we'll meet later.